Chapter 2: Membranes as Decision-Making Surfaces. The membrane is usually introduced as a boundary, and that description is correct as far as it goes, but it is still too passive to explain what membranes actually do. A membrane does not merely separate inside from outside. It interprets contact. It determines whether a molecule will be ignored, transported, resisted, internalized, amplified into a signal, or converted into a larger change in cellular behavior. In that sense, the membrane functions less like a wall and more like a decision surface. It is the place where the cell encounters the world and decides what that encounter means. Every tissue in the body depends on this process. Nutrients arrive, hormones circulate, inflammatory signals appear, microbes or microbial fragments make contact, mechanical force distorts the surface, temperature changes alter membrane behavior, electrical gradients shift, and the cell must continuously determine whether these inputs represent fuel, threat, instruction, noise, or damage. Those determinations are not made in abstraction. They are made through the physical properties and molecular organization of the membrane itself. This is why membrane biology belongs at the center of physiology rather than at its edge. The cell does not first understand a signal and then pass that signal through the membrane as a delivery route. The membrane is part of the interpretive mechanism. A receptor embedded in a membrane does not respond only because a ligand binds to it. It responds within a local field defined by lipid composition, cholesterol content, membrane thickness, surface charge, curvature, oxidative state, and the presence or absence of neighboring proteins. That local field changes the probability of receptor clustering, the stability of active conformations, the speed of internalization, the access of kinases and phosphatases, and the likelihood that a brief external event becomes a sustained intracellular response. What looks from the outside like a simple on-off signal is usually the end result of many smaller physical decisions built into the surface architecture. The membrane does not think in the psychological sense, but it continuously sorts inputs into biological consequences. This decision-making role becomes clearer once signaling is understood as a spatial event rather than a purely chemical one. Molecules do not act in empty space. They collide within structured environments. Receptors are not scattered randomly across a featureless sheet. They occupy domains, enter clusters, separate from other proteins, move into ordered regions, leave those regions, bind adapters, and participate in transient signaling assemblies whose formation depends on membrane conditions. A hormone signal can be strong in one membrane context and weak in another even when hormone concentration remains the same. An inflammatory trigger can be amplified or dampened depending on how membrane lipids shape receptor behavior and downstream enzyme access. A growth signal can persist too long if the surface organization that normally restrains it has been altered. These are not minor effects. They are part of how the cell decides what kind of state it is in and what it should do next. The concept of the membrane as a decision surface also explains why lipids influence behavior far beyond their caloric value. A change in membrane composition is not just a biochemical substitution, it is a change in interpretive architecture. Alter the balance of saturated, monounsaturated, and polyunsaturated fatty acids within phospholipids, and you alter the packing characteristics of the membrane. Alter packing, and you alter receptor mobility, microdomain formation, membrane permeability, mechanical resilience, and enzyme-substrate interactions. Change cholesterol distribution, and ordered regions may become more or less stable. Increase oxidative damage, and the membrane may become more disorganized, more permeable, and less capable of hosting precise signaling events. The cell then begins making different decisions from a same external world because the surface through which it reads that world has changed. This is one of the deepest reasons that nutrition, inflammation, oxidative stress, endocrine signaling, and chronic disease cannot be understood as separate conversations. They converge at the membrane. The immune system makes this especially obvious. An immune cell does not merely possess receptors for threat. It has to organize those receptors into functional patterns on the membrane surface, distinguish weak noise from true danger, assemble signaling complexes, change shape, move, engulf, secrete, and later deactivate or resolve. Every one of those steps depends on membrane behavior. If the membrane becomes compositionally distorted, oxidatively damaged, or overloaded with unstable signaling substrate, immune interpretation changes. Threat may be recognized too aggressively, too diffusely, or for too long. Activation may occur readily, while resolution remains incomplete. The cell may enter inflammatory states faster than it can leave them. This is not because immune cells have suddenly forgotten their purpose. It is because the surface through which they make decisions no longer supports clean discrimination. A body can therefore become chronically inflamed not only because inflammatory signals exist, but because its decision-making surfaces have become less precise. The nervous system depends on the same principle under different constraints. Neurons and glial cells must handle electrical signaling, receptor trafficking, synaptic vesicle fusion, local inflammatory surveillance, and rapid adaptation to incoming information. These processes require membranes that can support highly specific patterns of excitability and communication. A synaptic membrane must decide whether a neurotransmitter pulse should trigger a strong postsynaptic response, a weak one, a temporary change in receptor density, or a longer-term plastic adaptation. That decision is not made by neurotransmitter concentration alone. It depends on receptor state, membrane composition, channel behavior, scaffold stability, and local lipid organization. When membranes are altered, the nervous system may not fail dramatically at first. Instead, it becomes noisier, more reactive, less stable, or less efficient. Precision declines before catastrophe appears. The decision surface is still operating, but it is making lower quality decisions because its structure has changed. This framework also clarifies why chronic dysfunction often presents as dysregulation rather than simple deficiency. A person may have enough hormones in circulation, yet still show poor response. They may have immune activity that will not turn off, appetite signals that do not match energy state, stress responses that remain exaggerated after the stressor has passed, or inflammatory chemistry that persists long after the original insult. In many cases, the problem is framed in terms of too much or too little of a signaling molecule. Sometimes that is true. But often the more foundational issue is that the membrane surface interpreting the signal has become distorted. The system is not merely lacking input, it is misreading input. Decision quality has degraded at the interface layer. That leads to amplification where restraint was needed, resistance where responsiveness was needed, leakage where compartmental control was needed, and persistence where termination was needed. Once membranes are understood as decision-making surfaces, the subject of omega-3 expands naturally into a much larger argument. Omega-3 fatty acids are not interesting only because they can become anti-inflammatory metabolites or support a desirable ratio on paper. They matter because they enter the membrane and help shape the physical conditions under which cellular decisions are made. They influence substrate availability for future mediator synthesis, but they also alter membrane behavior before those mediators are ever produced. They can change how receptors cluster, how signals propagate, how inflammatory responses escalate, and whether resolution pathways have access to the structural material required to function. This does not mean omega-3 acts alone or solves everything. It means omega-3 belongs to the architecture of interpretation. It helps determine what kind of surface the cell is using to read its environment. The deeper message of this chapter is that biology does not decide through disembodied information. It decides through matter arranged into responsive surfaces. The membrane is where chemistry becomes meaning for the cell, is where contact becomes instruction, where instruction becomes activation or restraint, and where long-term structural inputs shape short-term behavior. Once that is seen clearly, many scattered topics begin to align. Receptor sensitivity, inflammation, insulin resistance, neurotransmission, immune tolerance, endothelial function, barrier integrity, and recovery capacity all begin to look less like isolated failures and more like different expressions of how well the body's decision-making surfaces are built. That is the level at which the lipid story becomes coherent, and it is the level required for understanding why membrane composition is never just background material. It is part of how the body decides what to do next. The cell membrane as an interface. The cell membrane is often described as a border, but border is too static a word for what it actually is. A true biological membrane is an interface. It is the active surface where the cell meets everything it does not fully control and must therefore evaluate, manage, or transform. Nutrients arrive at this surface. Hormones arrive at this surface. Mechanical stress reaches this surface. Immune signals, microbial fragments, toxins, electrical changes, osmotic shifts, and inflammatory mediators all reach this surface. The cell membrane is the first place where outside conditions become intracellular consequences. It does not merely separate two environments. It governs the terms of contact between them. That is what makes it an interface rather than a wrapper. The membrane allows the cell to remain distinct from its environment while still exchanging matter and information with it in a controlled way. Without that interface, the cell could neither preserve its identity nor participate in a larger organism. This interface function depends on more than the existence of a lipid bilayer. It depends on the membrane being built as a selective and responsive surface. Embedded within it are receptors, ion channels, transporters, docking proteins, adhesion molecules, enzymes, and structural anchors, all positioned inside a material environment that affects how they behave. A receptor only matters if the membrane can hold it in the right orientation, allow it to move when movement is required, cluster it when clustering is required, and connect it to intracellular signaling machinery once binding occurs. A transporter only works if the membrane can preserve the gradient that gives transport meaning. An ion channel only matters if the membrane can maintain the separation of charge and concentration that makes opening and closing consequential. The membrane, therefore, does not simply host molecular tools. It creates the conditions that make those tools usable. Because it is an interface, the membrane has to solve two problems at once. It has to defend the cell from indiscriminate mixing with the outside world, and it has to allow carefully selected exchange with that same world. If it becomes too permissive, the cell loses control over ions, water, metabolites, and signaling exposure. If it becomes too rigid or poorly responsive, transport becomes inefficient, signaling becomes blunt, and adaptation slows. The membrane has to remain stable without becoming inert. It has to remain dynamic without becoming leaky. That balance is one of the most important material challenges in biology, and lipid composition sits near the center of it. The kinds of fatty acids built into membrane phospholipids, the local cholesterol content, the presence of sphingolipids, the degree of oxidative injury, and the mechanical demands placed on the tissue all influence how well that interface performs. What the membrane is made from changes how it negotiates the outside world. This is why the membrane is not just a line between inside and outside, but a site of interpretation. The same external signal can produce different cellular outcomes depending on membrane state. A hormone may bind its receptor, but the intensity, duration, and downstream quality of that response depend partly on how the receptor is positioned within the membrane, what nearby lipids stabilize or destabilize it, and whether the surrounding surface supports clean signal transfer or introduces noise. The same nutrient may be absorbed efficiently in one membrane condition and poorly in another. The same inflammatory trigger may produce an exaggerated response in a disordered membrane and a more proportionate response in a stable one. The membrane does not think, but it determines how much meaning an external event is allowed to carry. That is why it behaves like an interface in the deepest sense. It is the point where contact is translated into consequence. This becomes even clearer when the membrane is viewed as a layered communication surface rather than a simple barrier. On its outer face, it presents receptors and adhesion structures to the extracellular environment. Within the bilayer, it organizes proteins in space, alters their mobility, and creates microdomains where certain interactions are more likely to occur. On its inner face, it anchors signaling machinery to the cytoskeleton and intracellular pathways that will execute the result of the encounter. An event at the outer surface, therefore, does not stay at the outer surface. It propagates through the membrane into the cell's internal architecture. A binding event can alter gene expression. A mechanical deformation can change ion flow. A change in membrane composition can alter endocytosis, exocytosis, motility, or immune recognition. The membrane is a translation layer between external conditions and internal action. This interface role is especially important in tissues that live under constant environmental exposure. Intestinal epithelial cells face food components, microbial products, bile acids, enzymes, and immune surveillance signals all at once. Their membranes must allow nutrient absorption while resisting uncontrolled entry of harmful material. Endothelial cells lining blood vessels must respond to circulating molecules, shear stress, immune signals, and redox changes while preserving vascular integrity. Immune cells must use their membranes to detect threat, distinguish self from non-self, form contact zones with target cells, and later disengage when the job is done. Neurons must receive neurotransmitters, maintain electrical gradients, and recycle signaling machinery at high speed without losing stability. In each case, the membrane is not an accessory to function. It is where function begins because it is where exchange with the world is structured rather than random. This also explains why changes in membrane composition can reshape physiology so broadly. When the interface changes, the quality of contact changes. If the membrane becomes more oxidized, permeability can shift and receptor behavior can degrade. If the lipid environment becomes less stable, signaling complexes may assemble less cleanly or disassemble improperly. If the membrane becomes overloaded with substrates that favor inflammatory mediator release under stress, immune signaling can become more easily amplified. If the membrane loses structural resilience, cells may become more vulnerable to mechanical or oxidative injury. These changes do not stay confined to one biochemical pathway because the membrane is upstream of many pathways at once. It is the platform through which multiple systems encounter their inputs. A degraded interface produces degraded interpretation across the organism. For that reason, the cell membrane should be understood as one of the body's primary control surfaces. It decides what gets in, what stays out, what gets sensed, what gets ignored, what gets transported, what gets stored, and what gets turned into action. It is not doing this alone, and is not doing it consciously, but it is doing it materially. Its composition, organization, and stability determine how precisely the cell can engage with its environment without being overwhelmed by it. Once that is understood, the importance of lipid architecture becomes much harder to ignore. Lipids are not merely passive ingredients in the membrane. They shape the interface itself, and by shaping the interface, they help shape the behavior of the cell, the tissue, and eventually the organism built from them. Receptors, fluidity, and signal interpretation. A receptor is often described as if it were a fixed lock waiting for the right key, but that image removes the receptor from the material surface that gives it meaning. Receptors do not float in empty space. They are embedded in membranes, and that embedding changes how they behave before any ligand ever arrives. The membrane affects receptor orientation, movement, clustering, conformational flexibility, access to signaling partners, and the rate at which activated receptors are internalized or recycled. This means signal interpretation begins before binding. The receptor is already being conditioned by the physical state of the membrane surrounding it. A hormone, neurotransmitter, cytokine, or growth factor may be present in circulation, but the effect of that signal still depends on whether the membrane is able to host a receptor in a way that preserves precision instead of noise. Fluidity sits near the center of this problem because membranes have to remain mobile enough to support signaling while stable enough to preserve structure. A membrane that is too rigid can restrict receptor movement, reduce the ability of receptors to cluster into active complexes, impair vesicle fusion, and interfere with conformational changes required for signal transfer. A membrane that is too disordered can create a different problem. Receptors may move excessively, signaling complexes may assemble too loosely or too unpredictably, permeability may rise, and the surface can become more vulnerable to oxidative injury and interpretive instability. Biology, therefore, does not aim for maximum fluidity or minimum fluidity. It aims for usable fluidity within a stable structural framework. The membrane has to allow motion without becoming incoherent. It has to permit adaptation without losing control of the surface on which that adaptation occurs. Receptors are highly sensitive to that balance because their function is not only chemical but spatial. Many receptors need to cluster after binding in order to amplify a signal. Others need to be segregated from inhibitory proteins before activation can proceed. Some require movement into cholesterol-rich or sphingolipid-rich membrane microdomains where the local environment supports certain protein-protein interactions. Some depend on rapid endocytosis after activation so the signal can be terminated or redirected. All of this is influenced by the material state of the membrane. The same receptor can generate a different biological response in two cells not because the receptor itself is genetically different, but because the lipid environment alters how that receptor behaves as part of a surface system. Signal interpretation is therefore partly a matter of membrane mechanics. The cell is not only asking whether a ligand bound, it is also determining what that binding event should be allowed to become. This is one reason membrane composition has such broad physiological consequences. Saturated fatty acids tend to promote tighter packing and structural firmness. Monounsaturated fats can preserve flexibility while maintaining relatively good stability. Polyunsaturated fats increase membrane mobility and can support dynamic signaling behavior, but they also introduce more chemical fragility because their double bonds are more easily oxidized. Cholesterol adds another layer by buffering membrane behavior, preventing excessive disorder in some contexts while preventing excessive rigidity in others. Sphingolipids contribute to order domains that help organize receptor activity and signal sorting. The membrane is therefore not simply more fluid or less fluid. It contains regions with different physical properties, and receptors behave differently depending on where they sit within that landscape. Signal quality emerges from the organization of this material terrain. The consequences of altered fluidity become obvious in endocrine and metabolic signaling. A cell may be exposed to insulin, thyroid hormone, cortisol, leptin, or other circulating messengers, yet still respond poorly if receptor behavior at the membrane has become distorted. This is often framed solely as receptor resistance or downstream signaling failure, but the membrane layer is frequently underappreciated. If receptors cannot cluster correctly, if local lipid domains are disorganized, if oxidative injury has altered the surrounding phospholipids, or if membrane turnover has produced a surface that no longer supports clean conformational change, the signal can be weakened, prolonged incorrectly, or interpreted with reduced fidelity. The body then behaves as though it has insufficient signaling even when the messenger itself is present. In that case, the problem is not absence of instruction, is degraded interpretation at the surface. The immune system depends on this same principle in a more dynamic setting. Immune receptors have to discriminate between background noise and meaningful threat. They often do this through threshold behavior, which depends on receptor density, clustering, membrane microdomain organization, and the speed with which signaling assemblies form and dissolve. A membrane that is compositionally unstable or excessively oxidized can lower the quality of that discrimination. Threat detection may become overactive, sustained too long, or poorly resolved. The cell may activate too easily and terminate too slowly. This is one pathway by which structural membrane problems become chronic inflammatory behavior. The immune cell is still receiving signals, but the surface through which it interprets those signals has become less reliable. A small input can begin to produce a larger and more persistent response than the original event justified. Neural signaling is equally dependent on membrane fluidity and receptor interpretation. Neurotransmitter receptors, ion channels, synaptic docking proteins, and vesicle release machinery all operate within membranes that must maintain fast dynamics without sacrificing precision. If fluidity drops too low, receptor mobility and vesicle fusion can become impaired. If disorder rises too high, electrical stability and signaling precision can begin to deteriorate. This does not always produce immediate failure. More often it produces noise. Synaptic timing becomes less exact. Recovery from signaling becomes less efficient. Excitability shifts. Inflammatory sensitivity in neural tissue may rise. The person experiences the downstream behavior as changes in mood, attention, resilience, sensitivity, or cognitive sharpness, but underneath those effects is a membrane-level problem of signal interpretation being carried out on altered surfaces. This also clarifies why omega-3 cannot be understood simply as a nutrient that turns inflammation up or down. EPA and DHA enter phospholipids and change membrane properties over time. That changes substrate availability for future signaling molecules, but it also changes the physical environment in which receptors and channels operate. In some tissues, this can improve flexibility, receptor behavior, or signal transduction. In other contexts, especially when oxidative protection is weak or membrane instability is already high, increasing highly unsaturated lipids without structural support may introduce vulnerability as well. The issue is never just whether a fatty acid is good in isolation. The issue is what kind of interpretive surface it helps build. Signal interpretation is only as good as the membrane through which the signal is being read. Receptors, fluidity, and signal interpretation therefore belong to the same system. A receptor is not a detached sensor. It is a protein embedded in matter, and that matter changes the meaning of the signal it receives. Fluidity is not a cosmetic feature of membrane. It is part of what determines whether receptors can move, cluster, change shape, terminate signals, and communicate with intracellular machinery at the right intensity and duration. When this relationship is stable, the cell reads its environment with high precision. When it is distorted, the same world begins to produce worse decisions. Hormones can be present but poorly felt. Immune triggers can be exaggerated. Neurotransmitters can become noisier. The membrane is not just where signals arrive. It is part of how signals become biologically real. How membrane composition alters reality. The body does not experience the world directly. It experiences the world through surfaces. Every nutrient, hormone, cytokine, neurotransmitter, mechanical force, temperature shift, microbial fragment, and oxidative stressor reaches the cell only by first encountering a membrane. That means the cell's version of reality is always filtered through composition. The membrane is not a neutral window. It is an active material layer that changes what can be sensed, how strongly it can be sensed, how long the signal persists, and whether the signal is translated into useful action or into distortion. Change the composition of the membrane, and the world the cell lives in changes with it, even if the external environment remains the same. Two cells exposed to the same molecule are not necessarily living in the same biological reality if their membranes are built differently. They will not read the same input the same way because the input is being processed through different physical conditions. This is one of the most important reasons composition cannot be treated as a minor biochemical detail. A membrane enriched in more stable lipid species behaves differently from one enriched in highly unsaturated, oxidation-prone material. A membrane with coherent cholesterol distribution behaves differently from one in which order domains are unstable or poorly maintained. A membrane with adequate phospholipid turnover and structural repair behaves differently from one that has accumulated damaged lipids faster than they can be removed. These differences alter permeability, charge behavior, thickness, flexibility, protein mobility, vesicle formation, and the probability that receptors and enzymes will encounter the right partners at the right time. The cell then begins drawing different conclusions from the same external event. A signal that should have been brief may linger. A stimulus that should have remained local may spread. A threat that should have been resolved may stay active because the surface conditions favor escalation more than closure. Reality, at the cellular level, is not just what arrives. It is what the membrane allows arrival to become. This helps explain why membrane composition alters not only signaling intensity, but signaling meaning. A hormone does not simply carry a message in pure form from the bloodstream into the cell. It meets a receptor embedded in a specific lipid environment, and that environment changes whether the receptor is properly folded, how easily it clusters, whether it enters an ordered membrane region, how effectively it couples to downstream proteins, and how rapidly the signal is terminated or internalized. The same hormone concentration can therefore produce different physiological outcomes depending on membrane state. The same principle applies to immune recognition. A receptor meant to detect danger may become oversensitive in a disordered membrane, less selective in an oxidized membrane, or unable to terminate correctly in a membrane that cannot reorganize cleanly once activation has occurred. What the cell interprets as danger, safety, abundance, scarcity, or injury is partly determined by the composition of the surface doing the interpretation. This is why chronic dysfunction often feels like the body has started misreading reality. A person becomes unusually reactive to food, stress, exertion, temperature, or inflammation. Hormonal signals seem present but ineffective. Appetite cues no longer match energy state. Immune activity activates too easily and turns off too slowly. Neural signaling becomes more erratic, less stable, or more easily overstimulated. These are usually described as separate forms of dysregulation, but many of them can be understood more clearly as interpretive problems emerging from altered membrane composition. The world reaching the body has not necessarily changed in proportion to the reaction. The body's surface architecture has changed, and with it the threshold, fidelity, and timing of biological response. In practical terms, the organism begins living in a noisier version of reality because its interfaces are no longer filtering and translating information with the same precision. The physical properties of different lipids are central to this effect. Saturated fatty acids tend to support tighter packing, lower permeability, and greater resistance to oxidative distortion, which can increase structural stability but also reduce mobility if present without balance. Monounsaturated fats often preserve fluid behavior while maintaining good resilience. Polyunsaturated fats expand flexibility and signaling range, but their double bonds increase susceptibility to peroxidation, which means they can enrich function under protected conditions and increase fragility under unprotected ones. Cholesterol acts as a structural buffer and organizer, modifying disorder in one region while preserving fluidity in another. Sphingolipids help create ordered membrane microdomains that shape receptor localization and signal sorting. None of these features are abstract biochemical trivia. They are the physical basis of interpretation. They determine whether the membrane behaves like a stable control surface or a chemically unstable field that distorts the signals passing through it. This is especially important because membrane composition is not static. It is continuously remodeled by diet, endogenous lipid metabolism, oxidative stress, inflammation, hormonal state, mitochondrial function, lipoprotein transport, phospholipid turnover, and tissue-specific demands. A person is not carrying one fixed membrane identity throughout life. Their surfaces are being rebuilt in real time, sometimes gradually, and sometimes under heavy metabolic pressure. This means reality at the cellular level is also moving. A tissue exposed to repeated oxidative injury may begin interpreting routine inputs as more threatening because its membrane environment has become easier to destabilize. A tissue repeatedly supplied with unstable lipids but insufficient antioxidant and repair support may develop high signaling volatility without durable control. Another tissue may become excessively rigid, less adaptive, and less capable of dynamic response. These are not merely changes in composition. They are changes in the rules by which the cell reads the world. The nervous system makes this especially clear. A neuron depends on membrane composition for excitability, receptor behavior, synaptic vesicle cycling, and recovery after stimulation. Change that composition and sensory thresholds change, mood regulation changes, attention changes, signal-to-noise ratio changes, and inflammatory sensitivity within neural tissue can change as well. The person experiences this as a psychological or cognitive shift, but part of what has changed is the membrane-level reality through which neural signals are being interpreted. The immune system follows the same logic through a different expression. Membrane composition changes how aggressively receptors assemble, how easily inflammatory cascades begin, how lipid mediator substrates are stored and released, and whether resolution can be organized once the initial response should end. The immune cell is not just fighting the world. It is reading the world through its own surface material, and when that material changes, its definition of threat changes with it. This is the level at which omega-3 becomes meaningful in a deeper way. Omega-3 fatty acids do not simply add a beneficial compound to the bloodstream. They alter membrane composition over time, and by altering membrane composition, they alter what kind of world the cell is inhabiting. They change the reservoir from which signaling molecules can later be generated, but they also change receptor environment, membrane curvature behavior, surface flexibility, and the probabilities of certain signaling events occurring. That does not make omega-3 universally corrective in every context. A highly unsaturated membrane that lacks oxidative protection can become vulnerable even while gaining signaling flexibility. A tissue overloaded with inflammatory competition may incorporate omega-3 without fully expressing its benefits. A person may require broader structural correction, not just one substrate change. But the principle remains the same. When membrane composition changes, reality changes for the cell. Seen clearly, this is one of the most important unifying ideas in biology. Cells do not respond to the world as detached processors of clean information. They respond through material surfaces that decide what counts as a signal, what counts as noise, what crosses the threshold for activation, and whether activation can later be resolved. Membrane composition therefore alters behavior at every level above it. It changes immune tone, hormonal sensitivity, neurotransmission, barrier control, mitochondrial efficiency, and stress tolerance because all of those processes depend on how the cell interprets contact. The membrane is the lens, and lipid composition changes the lens. Once that is understood, many forms of chronic dysfunction stop looking mysterious. The system is not always failing because the world is too hostile. Sometimes it is failing because the surface through which the world is being read has been rebuilt into something less precise, less stable, and less capable of telling the difference between signal and distortion. Lipid structure determines cellular behavior. Cells do not behave according to genes alone, and they do not behave according to signaling molecules alone. They behave through the material conditions in which genes are expressed and signals are interpreted. Lipid structure is one of the deepest of those conditions. It determines the quality of the membrane, the stability of compartments, the movement of receptors, the organization of microdomains, the permeability of surfaces, the vulnerability of those surfaces to oxidation, and the availability of stored fatty acid substrates that can later be turned into signaling molecules. Because of that, lipid structure does not merely support cellular behavior from the background. It helps change the lipid architecture of a cell, and you change what that cell can sense, how precisely it can respond, how strongly it reacts, how long it stays activated, and how well it returns to baseline once the event has passed. This becomes easiest to see when the membrane is treated as part of the decision system rather than as passive packaging. A receptor embedded in a well-built membrane behaves differently from the same receptor embedded in a membrane that is disordered, oxidized, overly permeable, or compositionally unstable. In one setting, ligand binding may lead to clean clustering, efficient downstream coupling, appropriate amplification, and timely signal termination. In another, the receptor may cluster poorly, overcluster, couple weakly, internalize incorrectly, or remain active longer than it should. The difference is not necessarily in the receptor protein itself. The difference is in the surface that holds it. The cell then behaves differently because its interpretive machinery is being run through a different physical environment. What appears from the outside as altered sensitivity, resistance, hyperreactivity, or dysregulation is often the behavioral expression of altered lipid structure. The same principle governs ion channels and transporters. Cellular excitability, nutrient uptake, waste export, osmotic control, calcium signaling, and electrochemical gradient maintenance all depend on proteins that must operate inside membranes with usable physical properties. If the lipid environment becomes too rigid, channel gating and transporter cycling may become mechanically impaired. If the membrane becomes too disordered or oxidatively damaged, permeability can rise in unwanted ways and charge separation can become harder to maintain cleanly. That means even basic cellular timing changes when lipid structure changes. The cell may depolarize more easily, recover more slowly, leak ions more readily, import substrates less selectively, or lose gradient-dependent control over its own chemistry. Behavior at that point is no longer being determined simply by which proteins are present. It is being determined by whether the membrane gives those proteins a stable platform on which to work. Lipid structure also determines behavior by shaping how the cell handles stress. A membrane built from highly oxidation-prone material behaves differently under inflammatory pressure than a membrane built from more stable material. Once reactive oxygen species rise, peroxidation can spread through vulnerable lipids, altering membrane integrity, damaging local proteins, changing permeability, and generating reactive breakdown products that further distort signaling. A cell with structurally resilient membranes can absorb a greater degree of metabolic or inflammatory stress before its behavior becomes chaotic. A cell with fragile membranes begins misfiring earlier. It becomes easier to activate, harder to stabilize, and slower to recover. This is why the same inflammatory load or oxidative burden can produce very different outcomes in different tissues or different people. Stress does not act on blank surfaces. It acts on built material, and the structure of that material determines how the cell behaves under pressure. Immune behavior is heavily shaped by this logic. Immune cells depend on membrane structure to assemble receptors, form immunological synapses, engulf targets, release signaling molecules, migrate through tissue, and later shift from activation to resolution. The ability to recognize a threat is only one part of the problem. The cell also has to regulate the magnitude and duration of the response. If membrane composition favors unstable signaling, inflammatory cascades can begin too easily and terminate too poorly. If lipid substrate pools are skewed toward mediators that intensify inflammation more readily than they support its resolution, immune behavior shifts further in that direction. The immune system then appears overreactive or chronically activated, but underneath that pattern is a structural issue. The cell is behaving according to the materials built into its surface and stored within its membranes. Inflammation is not being generated from nothing. It is being generated from architecture. Neural behavior follows the same rule through a different kind of membrane demand. Neurons require tightly controlled excitability, rapid vesicle fusion, receptor trafficking, synaptic plasticity, and membrane recovery after repeated firing. All of that depends on lipid structure. A membrane that is too rigid may slow receptor movement and vesicle dynamics. A membrane that is too unstable may increase noise, alter ion channel behavior, and reduce signaling precision. The result may not be immediate neurological failure. More often it is altered behavior at the system level, greater irritability, poorer signal-to-noise ratio, reduced resilience, unstable mood, diminished focus, altered sensory thresholds, or impaired recovery from stimulation. These effects are often described in purely chemical or psychological language, but the chemistry is still being executed through membranes. The neuron behaves the way its surface allows it to behave. Mitochondrial behavior is equally dependent on lipid structure, and this is often missed because metabolism is usually described in terms of fuel supply rather than machine integrity. The mitochondrion does not produce ADP simply because fatty acids, glucose, or other substrates are present. It produces ADP because its inner membrane can organize the electron transport chain, hold a proton gradient, maintain crystal structure, and prevent excessive proton leak. Cardiolipin and other membrane components play direct roles in stabilizing respiratory complexes and preserving the architecture required for oxidative phosphorylation. If mitochondrial lipid structure degrades, behavior changes. Electron transfer becomes less efficient. Reactive oxygen species may rise. ADP production becomes more expensive. Stress signaling increases. The cell then behaves like is underpowered, inflamed, or metabolically inflexible because the membrane architecture of its energy system no longer supports clean output. Fuel alone cannot fix that. The behavior is being determined by structure. This is also why lipid structure creates long time horizon effects that are often misunderstood. The body remodels membranes gradually through repeated dietary exposure, endogenous synthesis, lipoprotein transport, oxidative damage, repair, and phospholipid turnover. Cellular behavior therefore shifts over time as structure shifts. A person does not wake up one day with a completely different signaling system because of a single meal. But repeated incorporation of certain lipids, repeated exposure to oxidative stress, repeated inflammatory activation, and repeated failure of repair can slowly rebuild tissues into different behavioral states. Receptors may become less precise. Immune cells may become more triggerable. Mitochondria may become less efficient. Barriers may become more permissive. Neural tissue may become more reactive. What looks like a collection of unrelated chronic problems is often the accumulated behavioral output of slowly remodeled lipid architecture. This is the deeper reason omega-3, omega-6, saturated fat, cholesterol, phospholipids, and membrane turnover matter. They are not simply nutrients or risk factors floating above the body as abstract variables. They become part of the surfaces through which behavior emerges. Omega-3 can alter membrane flexibility, receptor environment, and the substrate pool for pro-resolving mediators. Arachidonic acid can provide substrate for potent inflammatory signaling. Saturated structures can increase stability and reduce oxidative fragility, though excessive rigidity in the wrong context can create different limitations. Cholesterol can organize and buffer membrane behavior. The point is not that one class of lipid always determines outcome in isolation. The point is that cellular behavior is built from the combined structural consequences of the lipid environment. The cell behaves according to what it is made from. Once this is understood, many physiological contradictions become easier to interpret. A signal can be present but not produce the expected effect because the membrane cannot read it well. A tissue can look inflamed not only because inflammatory triggers exist, but because its structural state amplifies those triggers and prevents clean resolution. A mitochondrion can have access to fuel and still generate poor energy because the membrane architecture required for conversion is degraded. A person can feel unstable across many systems at once because the surfaces governing those systems have been remodeled into a less coherent state. Lipid structure determines cellular behavior because behavior is not separate from structure. Is structure in motion, structure under stress, structure interpreting input, and structure deciding what the cell can become from one moment to the next?